Thank you for the opportunity to present expert reviews for ICHM7 assessment. I'm Alejandra Trejo, and I'm with Gilead Sciences. Today, I want to talk to you about ICHM7 and our assessment workflow for the evaluation of potential mutagenic impurities, and following that, the in-silico assessment and how do we use QSAR methodologies. I'll finalize with some case studies where I present the importance of the expert review. So the purpose of ICHM7 is to provide a practical framework so we can identify, categorize, qualified, and controlled mutagenic impurities. The aim is to limit any potential carcinogenicity risk. This applies to new drug substances and drug products during their clinical development and in resulting marketing applications, but it also applies to existing marketed products when there are changes to the synthesis which result in new impurities or increased impurity levels, when you have changes to the formulation composition or process which result in new degradation products or increased levels of existing degradation products. It also includes changes to the indication or dosing regime that would affect the acceptable cancer risk level. In cases where there's new in vitro or in vivo data on an impurity that shows mutagenicity risk, this also applies. In assessing the potential mutagenic impurities our assessment starts with assessing the synthetic route. We identify any potential impurities, and this includes starting materials, reagents, intermediates, any known impurities or degradants. This basically includes a literature search of public data, scientific publications, our company database, and the aim is to find mutagenicity and carcinogenicity data available so we can start classifying all these compounds into class 1, 2, and 5. This information is based on experimental data, and based on these results, a class 1 will be a compound that is a known mutagen carcinogen. That means that has a positive Ames assay data or other data that indicates that this is a mutagenic compound, and there are carcinogenicity studies available. A class 2 compound will probably have a positive Ames assay or other data that indicates that it's a mutagen, but there are no carcinogenicity studies available. And a class 5 compound will have sufficient experimental data to demonstrate that there is lack of mutagenicity or carcinogenicity. So we know that this is not an alerting compound. There are, however, a lot of compounds that will not have available data in the literature from our route that we are assessing. In these cases, we use the accuser assessment to classify as class 3, class 4, or class 5 compounds. According to ICHM7, we should be using two complementary methodologies in our QSAR assessment. One is an expert rule base. This is a qualitative methodology, and in this case, the substances are scanned for substructures that are known to be present in toxic structures, and through an inclusion and exclusion criteria, these match atoms and bonds to any substance with non-carcinogenicity and shows a prediction. In the case of the statistical based methodology, this is a quantitative methodology where the compound itself is divided in target compounds into substructures. These substructures then have a probability of being an Ames positive based on an analysis of a training set of compounds with known mutagenicity. So based on these two complementary methodologies, then the classification will result into an impurity into class 3, 4, or 5. A class 3 compound will be the one that has an alerting structure that is not related to the drug substance or any other intermediate or known compound with mutagenicity data. A class 4 compound will be that has an alerting structure. However, the same alert is in a drug substance or other compounds related to the drug substance or process intermediates, which have been tested and are non-mutagenic. And a class 5 are those compounds that have no structural alerts or alerting features, or there is enough data to demonstrate lack of mutagenicity or carcinogenicity. So I said in the previous slide that with an in-silico assessment alone, you could classify an impurity into a class 3, 4, or 5. However, in practice, this is not the case. There are several reasons why an expert review will be useful. One, which is very common, is to determine an outcome when results are out of domain, inconclusive, not in domain, or are inconsistent, meaning the statistical and the rule-based models will have different predictions. It is also useful when you need to improve that prediction. For example, in looking at the software, did the structures are interpreted correctly? Or there are other positive structures in the database that are representative of the functionality of the structure. Does the database have reasonable predictive power for the structure? Does that mean that does it have enough structures or enough related structures that we consider that this is truly a predictive result? So my first case illustrates the expert review in predicting an outcome when you encounter conflicting predictions. So in this case, for the indosol, the statistical based model shows predicted as positive, while the rules-based model predicted indosol as negative. So in this case, we would like to review the alerts and training structures in the statistical based model and to understand why that prediction is came to be. And at the same time, it's useful to look at similar compounds, either from the public literature or from other internal analogs, including the API. So in a review of the training structures for the statistical model, we find that we look at the positive training structures and the negative training structures, and it's very quickly apparent that the positive training structures contain in general secondary alerting features. As you can see, there's nitrous, there's amines, there's hydrazines, which are probably driving that positive result. In contrast, when we look at the negative training structures, you can notice that the same heterocycles, once they don't have that secondary alerting features, they are negative in mutagenicity assays. It's also, we also noticed that the indosol itself wasn't part of the training set. So looking at comparisons of the indosol and their presence as substructure or in the literature, indazole itself is a substructure of our API, which was already tested in an OECD bacterial reverse mutagenicity assay, and it was negative. There's also other available data. Indazole has been tested, and it was negative in the Salmonella typhimurium strains TA98 and TA100 without activation. So taking all this data into consideration, the fact that the secondary alerting features present in the positive training sets are likely driving that positive result, the comparison with the API, where indazole itself is a substructure of the API, and some mutagenicity data in the public literature, we conclude that indazole itself is negative, and then we classify it as class 5 for the ICHM7. Our second case is illustrates the not in domain. And this is, in my experience, a very common occurrence. There is a limited training sets in the databases, and not every single molecule will have all the uh, substructures available to make a prediction. So in our case, well, going back, ICHM7 wants to have the prediction of two methodologies, and a not in domain or inconclusive is not considered a prediction. So in our work process, we like to evaluate the molecules that are not in domain in the first statistical base method, in a second base statistical base method. And the reason why is because the different models have different training structures, and even if you have a not in domain in one, doesn't mean that a second model will not have some training sets that will allow a prediction. In this case, we did still have a not in domain prediction in the second. The reason why in both models this particular compound was not in domain was due to the unknown fragment hydroxymethane sulfonic acid. However, in spite of some of these structures not being present in the database, looking at public literature data, we found that this unknown fragment actually is contained in several structures that have Ames data, and they were negative in different strains of Salmonella typhimurium and some in E. coli as well. So based on this data, we conclude that although this is not in domain in the statistical base models, this unknown fragment is present in several structures that had negative mutagenicity results. Therefore, we consider that these molecules should be predicted negative, and the ICHM7 classification should be class 5. The case 3 illustrates what happened whether or not you have a prediction, and in this case, according to ICHM7, you have a prediction in both QSAR methodologies. This particular compound is negative in both the statistical and the rule space, but we still like to review the training structures and look for data outside the QSAR predictions. So looking at the training structures, we find that the pieces, which is the cyclopropane, is present in several structures in the database, as well as the carbonyl alkyl. But one thing that was noticed was that even though there was different structures, the ones that have an aldehyde itself were big structures. There was definitely a bigger group than our cyclopropylaldehyde. Comparing this with some literature search, we found that close analogs like acetaldehyde and formaldehyde, which are very small aldehydes, were known mutagens and potentially carcinogenic. So in spite of having a negative prediction in our QSAR, we felt that the known similarity, the similarity with some known mutagenic compounds should be taken into account, and we put this compound as positive and as a class 3, and this should be reviewed and assessed further to truly ensure that this compound is negative before we move forward with a negative prediction. The case 4, again, we have a conflictive result from the statistical and the rules-based. I want to illustrate here that a similarity with a different compound can be useful. The alerting feature in this case was that alkyl halide in the rules-based model. However, we had an analog within our database, and that was very similar. The only difference between these two compounds was the amine was alkylated, and this alkyl didn't have any alerting feature. We compare both compounds in the QSAR, and they both have the same alerting features highlighted. Our analog, however, had been tested in the Ames assay, and it was negative. So based on the similarity to a close analog with negative Ames results, our expert review predicted this particular structure as a negative, and it was an ICHM7 classified as class 4. So in summary, I hope I show you our hazard assessment process. How do we use it to classify potential mutagenic impurities? What is our process to use QSAR methodologies? The fact that the ICHM7 recommends to use two, the rule-based and the statistical base, and the importance of the expert review of that QSAR results to provide additional evidence to support the relevance of any positive or negative predictions, and a rationale to determine a final decision on conflicting or inconclusive predictions. And with this, I want to thank you for your attention. And if you have any questions that MultiCASE cannot field for me, I would be very happy to answer these through email or any other communication. Thank you.
Okay, so that concludes Alejandra's presentation. And instead of jumping right into questions and answers, I'd like to go ahead and move into the second presentation, and then we can have a extensive Q&A at the end. So I'd like to introduce our next presenter, who, sorry, give me one second. Okay. Who is actually on call with us today and will be available to answer questions and answers, as I mentioned at the beginning of the session. Dr. Sebastian Joseph is a veterinarian and a certified toxicologist from the Diplomat of the American Board of Toxicology and a European registered toxicologist, and is currently working as the director and principal consultant for development of consulting and scientific affairs at Pharmalex, India. Dr. Sebastian has close to 14 years of industrial experience that includes drug discovery, non-clinical development of drugs and vaccines, experimental toxicology and pharmacology, good laboratory practices, computational toxicology, human health, risk assessment, environmental risk assessment, and medical writing. In his past assignments, he's established and validated a number of in vitro and in vivo safety pharmacology models, and contributed significantly in establishing an integrated safety pharmacology facility. He has extensive knowledge of OECD principles of GLP, its implementation and application in a regulatory environment for the safety evaluation of pharmaceuticals. He functioned as a study director for toxicology and safety pharmacology studies, and also as a study monitor for outsourcing key investigational new drug application enabling studies. Dr. Sebastian is currently involved in strategic development and consulting services in pharmaceutical product development with special reference to non-clinical and toxicological requirements, non-clinical evaluation strategy in support of IND and NDA, risk assessment of pharmaceutical active ingredients and excipients, impurity qualification of APIs and drug products, risk assessment and derivation of permitted daily exposure and occupational exposure limits in support of GMP manufacturing, environmental risk assessment of pharmaceuticals, providing expert review of investigators' brochure, non-clinical and clinical overview of CTD modules, author and reviewer briefing book for regulatory consultation, response to regulatory queries as appropriate, and toxicological risk assessment and safety evaluation of both cosmetic ingredients and products, medical devices, and consumer products. So clearly, Dr. Sebastian is a wealth of knowledge on many subjects, and I'm very pleased to begin his presentation on mutagenicity evaluation.
Thank you, Gianna, for your brief introduction. Welcome you all for this brief webinar. I will be talking about mutagenicity evaluation beyond QSAR. So in this presentation, I will be talking about ICH M7 hazard evaluation strategy and control limits, and then how to derive limits for cohort of concern, N-nitroso compounds, that's nitrosamines, and a little bit background about these compounds, and then how to set limits for nitrosamines, those who are not having carcinogenicity data. And in the form of case study, I will be taking you through derivation of acceptable intake for N-nitroso ethyl propyl amine. The fundamental principle of ICH M7 is that genotoxic carcinogens are mutagens, and it is assumed that mutagenicity will invariably lead to carcinogenicity. The assessment of hazard related to mutagenicity and carcinogenicity is initiated with a comprehensive literature search to obtain data pertaining to mutagenicity and carcinogenicity. And if you find that impurity is not a mutagen, then you typically assign the impurity into class 5. If the impurity is mutagenic and possesses carcinogenicity potency data, and it's found to be a known carcinogen, you will typically assign the impurity into class 1. In some cases, you will see that impurity is mutagen, but you don't know really whether this is going to be carcinogenic or not. So in those situations, the impurity will typically fall under class 2. Now, when you do a literature search, in most of the situations for impurities, you probably will end up in no data. And that's where exactly you are going with another approach, that's quantitative structure activity relationship. As per ICH 7, typically QSAR is to be done using two complementary tools, one statistical based and the other expert knowledge based tool. And this should be followed by an expert review that will end up classifying the impurity into either class 3, 4, or 5. If the predictions are concluded to be not mutagenic, typically the impurity will be a class 5. In cases where impurity is showing structural alerts and those alerts are not shared with the API, then the impurity will be called mutagenic and assigned to class 3. In some cases, structural alerts are present. However, these same alerts are shared by the API or other related compounds and which are tested to be Ames negative. And those compounds typically will be assigned class 4. So this way, impurities are assigned to one of the 5 classes of ICH-M7. Now, the next question is, at what level we need to control these impurities? For impurities which are not mutagenic, that's class 4 and class 5, the limits are guided by ICH Q3A and Q3B guidelines. And for class 3 and class 2, the default approach is to control the concentration of the impurity in the API or the drug product at concentration that's equivalent to a daily exposure of 1.5 microgram per day. Now, I keep hearing from people saying that my impurity is a known carcinogen and therefore I'm going to control it at the TTC of 1.5 microgram. This approach is not correct. When you know that your compound is carcinogen and you have carcinogenicity data, TTC approach is not valid. So we will talk about what approach should be taken in those situations little later. Now, while TTC is a limit that can be followed by the companies and that can certainly help companies to decide on what limit should be fixed in their product and API, TTC is not always applicable. There are exceptions. There are certain compounds belonging to certain structural class. They are known to have high mutagenic carcinogenicity potency. So these compounds are called cohort of concern as per ICH-M7. For example, aflatoxin-like compounds, N-nitroso compounds, and alkyl azoxy compounds. For these class of compounds, the TTC is not applicable. And so what will be the next approach? How do we control, how do we define the limit for these impurities? We will talk about this process taking an example of an N-nitroso compound. Now, N-nitroso compounds also can be called as nitrosamines, are unwelcome guests in the pharmaceutical industry. Since July 2018, number of drug products were found to contain trace amounts of different nitrosamines in the drug products that led to withdrawal of number of batches of many drug products manufactured by different companies. Valsartan, losartan, ranitidine, metformin, they are just few examples. Now, what are these nitrosamines? So nitrosamines are chemicals that contain a nitroso group. With respect to N-nitrosamines, this nitroso group is attached to a nitrogen. And the figure that's given in the right corner is a general scheme of a nitrosamine compound in which R1 and R2 could be a simple hydrogen, it could be alkyl or allyl side chains. And these nitrosamines are formed in the presence of secondary or tertiary amine and nitrate, especially under acidic pH conditions. The further investigations found that these nitrosamines in those drug products were formed or originated from reagents, catalysts, solvents used in the manufacturing, under certain manufacturing conditions. Now the question is, why are we talking about nitrosamine? Why they are so concerned? Now, nitrosamines are highly DNA reactive. That means, they cause mutation. They are potent mutagenic carcinogens. Now, about 90% of the 300 nitrosamines tested were deemed to be carcinogenic, and therefore nitrosamines are associated with a high risk of cancer. And International Agency for Research on Cancer has classified nitrosamines as probable human carcinogen, that's category 2A. Now, how do we handle nitrosamine in the drug products? As a response to the observation that many drug products contain nitrosamine, industry and the regulators worked and explored ways how this can be handled. And the EMA has come up with a detailed guidance as to what manufacturers should do in terms of identifying this risk and controlling this issue. It starts with performing a risk assessment of their manufacturing process. And every manufacturer, drug manufacturer, is supposed to complete this risk assessment by end of this year, that's December 2020. And when the risk assessment identifies that there is a risk of formation of nitrosamine, this is supposed to be confirmed by way of analytical testing. And if nitrosamines are found to be present, then efforts should be put in to control that and limit their level in the drug product and API to an acceptable level or less than acceptable intake that needs to be derived using toxicological methods. Now, this acceptable intake or AI is based on the principle of excess cancer risk. So that means the mutagenicity or DNA reactivity follows a non-threshold dose response. So that means there is no dose that is considered free of cancer risk. Theoretically speaking, even a single molecule of a nitrosamine, for that matter, any mutagenic DNA reactive compound, is associated with some level of cancer risk. And therefore, it's not possible to identify a dose that's completely free of cancer risk. And that's the reason a methodology called excess cancer risk is adopted. As per ICH-M7, for pharmaceuticals, cancer incidence of 1 in 1 lakh population exposed is considered acceptable, 1 in 1 lakh. And this is done by linear extrapolation of cancer potency data, further down, extrapolated downwards, and to a dose that's not going to increase the cancer risk more than 1:1 lakh. And this cancer potency data is obtained from public literature. Now, the challenge is that when you look for public literature for nitrosamine compounds, or for that matter, any mutagenic compound, for impurities, most likely that you encounter a problem that there is no adequate data available. There is no adequate cancer potency data that can serve as the basis for derivation of AI. Then how do we do that? What is the solution for this? ICH-M7 gives a provision. In chapter 7.5, it says a case-by-case approach using, for example, carcinogenicity data from closely related structures, if available, should usually be developed to justify acceptable intake. Carcinogenicity data from closely related structures. EMA Q&A document on nitrosamine states if the available carcinogenicity and genotoxicity data are not considered sufficiently robust or are not available, expert structure activity relationship analysis should be used to derive the interim limit. So essentially, for those compounds where adequate data is not there, we are going to follow surrogate approach or AI derivation using data of surrogate compounds. Now, in this slide, what you see here is a general scheme of deriving acceptable intake for N-nitroso compounds or nitrosamine. You will first start with a comprehensive literature search covering both medical, toxicological, and clinical safety data and general databases, and try to retrieve relevant carcinogenicity potency data. If adequate data is there, you will derive an AI using linear extrapolation method. If there are no data, then you will review the structural features of your impurity in detail and try to identify chemical structures that are related to your impurity. Then again repeat the literature search to retrieve cancer potency data and other toxicological data for those analogs. Then whichever analogs having adequate data will be further screened down, selected. Then a detailed review of cancer potency data will be performed. A suitable cancer potency endpoint will be selected and AI will be derived. Now, here I'm going to talk about a case study in which AI is derived for N-nitroso-ethyl propylamine or NEPA. So when we started this work, we did a literature search and we found that there's no data. And then we did a detailed review of the structural features. On the left hand side, what you see is a general structural scheme of N-nitrosamines, typically, as I mentioned, nitroso group and then R1 and R2. On the right hand side, what you see is the structure of our impurity. R1 in this case is ethyl, R2 is propyl, and then here we have nitroso group. So essentially this impurity contains two linear alkyl chains, ethyl and propyl. Now we couldn't find any data including mutagenicity data. Although it's expected that they are mutagenic, we did a QSAR evaluation using case ultra, both statistical as well as expert knowledge based modules. And then the system predicted that this impurity to be positive for mutagenicity with call confidence very high, which is not unexpected given the presence of nitroso groups in this impurity. Now, another reason why we want to do this prediction is that case-ultra database, has structures that contains data, and these structures which are related to our query compound. So it gives the prediction, it identifies the alerts, and it also gives details of structurally related chemicals or analog compound, and it also gives to what extent they are similar with the query compound. And so in order to understand more about analog and structural features, we did this case ultra prediction. And in the next step, we searched analogs in the ChemID database with a cutoff similarity criteria of 70% or more. There were 28 structural analogs or 28 hits, and we did the literature review again for these chemicals, and we could get data, relevant toxicology data only for 8 of them. And what you see in this slide and in the next slide is the information pertaining to these 8 analogs. There were, out of 8, there were 4 compounds which had similarity of around 80 to 87%, while the rest of them were between 70 and 75%. So if you look at closely our impurity structure, it's actually C2N3. Essentially, we are just trying to figure out the chain length, R1 and R2, and then compare it with the analog structures. So as I said, there were only, out of the 8 compounds, only 4 had cancer potency data, and therefore we narrowed down to those 4: structure 1, 4, 7, and 8. And a close look at the TD50 values gives an impression that, you know, as the chain length increases, the potency comes down. So that's one information that we have made use in the selection of the suitable analog. Accordingly, compound number 1 and 4 were found to be, the most probable choice, and compound 7 and 8, slightly higher chain length, and similarity is also low, and therefore, we had to select compound either 1 or 4. For compound 4, the TD50 value is lower than compound 1, and as we know that, chain length may have a bearing on the cancer potency, so we finally selected compound 4 as the most suitable surrogate for our impurity. Now, just to mention what were all the criteria in selecting and rejecting compounds. So one is certainly lack of toxicity data. We straight away reject them. Presence of additional groups, aldehyde, alcohol, or other longer chains. So they were, rejected in the first place. The selections were, interestingly, all of them, as expected, they were mutagenic. And as I said, only four of them had TD50 value. And it is reported that, metabolism of this kind of a compound, follow a particular pattern, as mentioned here, the reference. This is initiated by cytochrome P450 dependent enzyme system. It's essentially hydroxylation of alkyl chain. And then subsequently, hydroxylation at the alpha carbon is thought to form an unstable intermediate, leading to the formation of diazonium ion, which can react with the DNA and lead to DNA alkylation. So the analog and the impurity are expected to follow the same or similar metabolic pattern. And finally, we narrowed down to structure 4. This is a bit more about the metabolism. I'm not going to the details of that. Yeah, so here in this table, what you see are the compiled information, cancer potency data for the surrogate compound. Carcinogenicity data were available for both rodents, that's rat, and also primates, that's monkey and bush baby. Although one of the study in cynomolgus monkey was giving the most conservative TD50 value, given the limitations of the monkey study, because monkey studies, the design was not robust. Either animal numbers in each group were too small, or number of doses tested in each studies were very few, or dose spacing was too high, and overall inconsistency between the studies. When compared to monkey study, rat studies were robust. 8 independent studies were performed and 15 doses were tested, ranging from 0.001 milligram per kg per day to 1.15 milligram per kg per day, and duration more than 2 years. That's typically needed as per ICH-S1. So therefore, we finally selected the rat cancer potency data of 0.0265 of the surrogate compound to derive the AI. Now, as I mentioned, the methodology is a linear extrapolation from a starting point, or that starting point is called point of departure. You can use two options as a starting point. One is TD50, that's median tumor dose, or dose of the compound that's causing tumors in 50% of animals. Or you can go with BMDL10, that's benchmark dose lower bound confidence limit. That's the lowest dose, which is 95% certain to cause no more than 10% cancer incidence in rodents. So you can use any of these two. We derive the values, and essentially, if you are using a TD50, you need to divide it by 50,000, and if it is a BMDL10, you need to divide it by 10,000. So in the case of this compound, TD50 was 0.0265 mg per kg. When divided by 50,000, you have a value of 0.53 nanogram per kg per day, which for an adult of 50 kg standard weight, this corresponds to an acceptable intake of 26.5 nanogram per day. So this is how we derive an acceptable intake of 26.5 nanogram per day for the nitrosamine impurity N-nitroso-ethyl propylamine, which has no toxicology data with respect to carcinogenicity. And using this acceptable intake and taking into consideration the maximum therapeutic dose of the drug product, you will be able to define the acceptable concentration or specification limit in terms of ppm or percentage, which can be used as a guidance while developing product. Yeah, so this is all I wanted to present. Thank you all for your patient hearing.
Okay, thank you, Sebastian, for such an informative presentation. I'd now like to open it up to questions. So I'd like to ask our panelists to go ahead and be prepared to answer some questions. It looks like no one has their hands raised at the moment, so I'm just going to go ahead and start scanning the questions that were raised during the presentation. So Sebastian, if you're still there, hopefully you are. Rustam, Suman?
Yes, I'm here.
I'm here too.
Okay, okay.
I think Sebastian's microphone is grayed out somewhere.
Okay. So one question that came in was, sorry, let me get, so what is the toxicological approach when a compound is mutagenic but found to be non-carcinogenic, and how do you classify this impurity as per ICHM7?
That's an interesting question. That's this hits what we call a gray area. It's neither class 1 or class 2 because it's non-carcinogenic. And it would be class 2 if we did not know about carcinogenicity status. As far as I remember, class 2 is non-mutagen with unknown carcinogenicity concerns, and we know that it's non-carcinogenic. It's not class 5 either, because again, it's non-mutagen. I would say you just go by the mutagenicity first. So if it is a mutagen, you call it class 2, and then you refer to the carcinogenicity studies. Of course, they need to be expert reviewed if they're proper, and then you downgrade your call. I sincerely don't know what class you can call it, because there is no class for this in ICHM7 guidance. That means you need to create your own definition and explain it properly. Downgraded class 5, negative overall for carcinogenicity concern. And that's probably what you can refer to as a clinically irrelevant mutagen. But again, there is no formal class for this.
Okay, thanks, Rustam. So another question coming in is, so In-silico QSAR is using mostly Ames study data, but when you have a known mechanism which produces only positive results in non-bacterial mutagenicity assays, then how do you interpret those QSARs, as mostly it uses Ames bacterial data?
I'm not sure I got the question correctly. So yes,
Yeah,
Silico is using Ames data. Yes, that I can confirm. So Man, can you answer that?
can you repeat it, Gianna? It was the sound was kind of breaking off at my end.
Oh,
Can you
okay,
please?
sorry about that.
That's fine.
So In-silico QSAR is using mostly Ames study data, but when you have a known mechanism which produces only positive results in non-bacterial mutagenicity assays, how do you interpret those in the QSAR, as mostly it uses Ames bacterial data?
Well, if we look into the ICH-M7 guideline, there is in the class 2 assay, it says bacterial mutagenicity positive or other relevant DNA reactive positive data. So if it is known to cause mutagenicity by other mechanisms, that should be considered as well. Um, so bacterial mutagenicity should be considered as one of the ways, and one of the major ways to determine something. And we built QSARs using those bacterial mutagenicity data because that is available um, in large quantities, and it is the most known way to assess mutagenicity. But if there is some other data available for which there is no QSAR, but suppose you have you know that this compound causes mutagenicity by some other mechanism, that has to be considered. And if you know that that will cause mutagenicity or carcinogenicity, it should be classified as a class 2 compound, because that's exactly written in the ICH-M7 guideline. They say that bacterial mutagenicity positive or other relevant positive DNA reactivity data, including in vivo gene mutation like clusterogenicity and other kinds of things. Okay, that's what is needed.
Okay, thanks, Saman.
Hmm.
So another question coming in is, so use of surrogates when compound-specific data is not available, how much is appropriate? And given that there are various dissimilarities between the query compound and the surrogate, how do you handle that situation?
Yeah, it's a very important question, and it comes very regularly. Like how to actually consider the surrogates, or as we call analogs. And my reasoning behind it always is that it depends on the compound that you are trying to find an answer for. So, and that's the query compound. So, it depends on the structural features associated around the alert. And if you should be able to find analogs that show the same type of structural features, particularly around the alert. It's not depend on the overall structural similarity of the analog or the surrogate with your query compound, but it depends on the kind of structural features that are present around your alert that is identified in the query chemical. So, for example, suppose your query chemical is an aromatic amine and it has a fluoro group at the meta position in a ring, So aromatic amine, and there is a meta position halogen chlorine. Now you should be able to get analogs that contain same type of amine, aromatic amine, with a fluorine at the meta position. And another factor that has to be considered at the same time, that those surrogates should not have any other groups that can cause mutagenicity by themselves. Say, for example, your analog contains the same aromatic amine and a meta-position fluoro group, but it has a hydrazine or a epoxide at a different place. So that is not going to be a very good analog to support your decision. So these are the two factors that needs to be considered to select a surrogate. First is that it should show the same type of structural features around the alert. And second thing is that it should not contain other toxicological functional groups that can cause mutagenicity by themselves. Okay. Go ahead, Gianna.
Okay, yes. I was just unmuting Dr. Joseph, who I think we should be able to hear now. Dr. Joseph?
Hi,
Hello?
Diana. Yes.
Hi.
You are able to hear me?
Yes.
Yes.
Okay, yeah, that's good.
Okay,
Yeah,
so.
it was muted.
Oh, I see.
I was not having the right to unmute it.
Oh,
Okay.
no. Okay.
You're good.
Sorry about that. Well, I have some questions for you. So let's see. In case of the monkey species, could you see a different species could affect the results as like we see in rats?
Yes, certainly. There can be considerable species difference in the qualitative cancer potency and also the quantitative, so in some cases, one species you may have cancer in one organ, whereas in the other species you may have a cancer in another organ, and also the doses could be different. Generally, monkeys, one of the limitation is that there are not many studies with monkeys, and traditionally the cancer studies are driven by ICH-S1, which recommends rodents because it's simple to perform, and therefore you have more experience with the rodent carcinogenesis and rodent mechanism of carcinogenesis, and therefore you will usually find that rodent data is more robust compared to monkeys, and that's the reason why in most of the cases typically, the risk assessor tend to pick rat data.
Okay, thank you. Another question coming in is, in case of surrogates, recent reports say that due to chirality, the body behaves differently, and normally there's a consideration of kinetics, but usually we tend to ignore dynamics. So is it not high time to modify strategy, as no one still has an idea of how body receptors are behaving to analogs due to optical isomerism as they get separated during processing in the body? So more of a comment than a question. But
yeah, I agree somewhat that the pharmacodynamics and pharmacokinetics should be considered, and sure, if you have enough knowledge about the surrogate that may not be mutagenic due to certain differences in the pharmacodynamics or pharmacokinetics, it should be considered right at the expert review process.
okay,
Yeah.
we do have. Oh, sorry. Go ahead, Dr. Joseph. Go ahead.
Just want to add that as the risk assessors, we always want to work with the data that's available for the impurity of concern. It's not that we want to utilize the surrogate compounds, but we are in an unfortunate situation that we have to make use, because in most cases data is not available, and therefore there is some compromise it's made always when you're working with surrogate, and usually we tend to be conservative.
Yeah, absolutely. I agree. And majority of the time, we also as we go forward in the future, we keep adding new and new details of the compound that we put into our database, which come up during an analysis, like similar analogs and stuff. And depending on the situation, I consider I'm not a toxicologist, but as far as stuff that I've seen so far, toxicology is a science of exceptions. And anytime you see something that is out of the ordinary, it should be looked into in more detail.
Hmm. So I have a few hands raised. I'm going to go ahead and unmute Malikaranjan. He's had his hand up for a while. So, sir, be prepared to be unmuted to ask your question. Hello, sir. Can you hear us? Okay, moving on. Anat Kulkarni, I see you have your hand raised. I'm going to go ahead and unmute you. Hello?
Dr. Kulkarni, can you hear us?
Okay, Nurali Patel also has their hand raised. Okay, Ravindra, you have your hand raised. I'm going to go ahead and unmute you. Hello, Ravindra, can you hear us? Hello?
Yeah, hello.
Hi.
Yeah, thank you
Go
very
ahead
much.
with your question.
Yeah. So I have a couple of questions. I posted it, but I want to ask one question on nitrosamine. So when the derived value for a nitrosamine impurity is coming more than the regular TTC level, which is genotoxic, because as we see, according to guideline, aflatoxins are said to be more toxic than regular PGIs. So when it comes, the derived value is more than TTC, is it allowed to follow?
Sebastian,
Yes,
probably should.
Thank you, Ravindra. That's a very good question. Default numbers or default limits are all, default, just to make our life easy in the absence of data. However, when you have carcinogenicity data, which is reliable and of a high quality, then, and you use that data to derive a compound specific limit, then, it's perfectly acceptable to follow that limit derived based on the compound specific data rather than the TTC. So compound specific data always overrules the TTC, provided the quality of the data. That's very, very important. Quality of the studies should be of highest standard as per the guidelines. So if that's met, yes, then you can go ahead with the compound specific limit.
Okay, clear. Thank you very much. And another question is when the nitrosamine impurity, when we see the similarity structures, so is there any criteria defined for the similarity definition? So how much percentage it has to be similar? Is there any tool to assess the similarity?
Yeah, I
Go ahead, Sebastian.
I don't think there is any, at least to my knowledge, there is any percentage defined. My understanding is that, the more the percentage or more closer the surrogate to our impurity, I think data is likely to be more reliable. But I think there is no percentage numbers defined.
Yes.
Suman, you have anything to add?
Yeah, I totally agree with you. There is no clear-cut percentage or similarity value has been defined to be used, because again, the reason is that the compound that you are working with, your query compound, may have a certain part which contains the alert. So it is important to use the most similar compounds first, and then you have to find out of those most similar which one actually is very close to the structural features associated with the alert that you are seeing in your query compound. That is the way to go. Instead of looking into the absolute values of the similarity measure between your query compound and the surrogates, So it's always an exercise to find the reasons of toxicity instead of focusing on particular values. Sometimes I have seen that the surrogate is just have a similarity of 0.4, but the good thing is that the structural features around the alert is very similar to the query compound. Then it is a good one. Only thing that we need to consider at that point, and that actually brings in the pharmacodynamics and pharmacokinetics, is that suppose the structural features around the alert is very similar. I mean, the groups are same, electron withdrawing or electron donating or steric or whatever it is, but the other part of the chemical contains a huge polar group, like some sugar moieties or something, that would actually change the surrogate's pharmacokinetics a great deal. And then that's not a good analog to consider. So this is another factor that needs to be considered. Okay.
Yeah, thank you. So can I take one more question?
Sure.
Thank you. Regarding this nitrosamine impurities, as you know, industry is screening all their APIs for their presence due to the implications posed by the regulators. So when it comes to the synthetic possibility and the practical possibility, so there is a lot of challenges seen when we are assessing the risk assessment. So during this, when the impurity is theoretically possible and is practically, it is difficult to not possible to make. So how do we, and it is not reported in literature, how do we manage it with respect to control?
Yeah, so that
is it
goes
okay
into the, yeah, please.
Yeah, is it okay to give a rationale that synthetically it is not possible?
Yeah, absolutely. Like, if there is a good reason that the impurity is not going to end up in the final product, then it is out of question for ICHM7 point of view, because the guideline says the possible impurities. So if the impurity is not really possible for getting into the final product, and you can show the reasons that it has a high solubility, it has been washed properly, and the final the spectrophotometry analysis or whatever you do to detect those things do not show any kind of indication that to show that this impurity is ending up in the final product, then you can definitely put that into your arguments that this impurity is theoretically possible, but practically it's never going to end up in the final product because of this and this and this reason. I think that is quite reasonable explanation.
Yes, thank you very much. I'm done.
Sure.
Okay, great. So this is semi-related. It's if a molecule is clastogenic but not mutagenic, how do you address such molecules in this situation?
As far as I'm not very knowledgeable about this thing, but as far as the ICHM7 guidelines is right in front of me, and it says that in the class 2 definition, it says clearly known mutagens with unknown carcinogenic potential, and in the bracket it says bacterial mutagenicity positive, and with a footnote that all other relevant positive DNA reactivity data, e.g., for example, in vivo gene mutation studies. So if a compound is clastogenic and you have a proper experimental data for that, that is actually put it into the class 2 compounds. But you should have somewhat more details about that thing. Like, bacterial mutagenicity is very direct, and it is the most common indication of mutagenicity. But I think in the ICHM7 guideline, it clearly gives an indication that other types of mutagenicity gene mutation data is also valid.
So that would also include corresponding metabolites.
Yeah, particularly metabolites, and particularly it will become even more important if the clastogenicity is positive. If it is negative, it may not say that everything is fine. But if it is positive, then you can think that this is a mutagen. And as far as the metabolite is concerned, now suppose you have a drug that is known to be fine. I mean, it's non-mutagen, and it has some metabolite that is coming up with some sort of mutagenicity positive data. But since this drug has already been approved and it has been in use for some time, those metabolites should be considered safe. But if you find a new metabolite and you can see that there is a new group that is present in it, but again, the same thing happens, if it is, you should show that there is a possibility of this metabolite and there is a change in dosage or some other types of administration, route of administration or change in that will change the metabolism pathway. There is a possibility, then that has to be considered. So for example, if you are giving a drug through as a tablet, and now the tablet obviously it goes through GI and metabolized through liver and all these things, and so then there is no concern even if you know that there is a metabolite which is positive. But if that drug now is given through some other route, say injection, and there is a new possibility of metabolism, and you know that there is a new metabolite possible, that has to be given an extra consideration.
Can I make a comment?
Sure.
If I understood that question correctly, the question was compound is carcinogenic, but it's not mutagenic. So that happens. So that means that carcinogen is a non-mutagenic carcinogen.
Yes.
Or the carcinogenicity is due to non-genotoxic mechanism.
Yeah.
And for a non-mutagenic or a non-DNA reactive mechanism of carcinogenicity, it is assumed that those carcinogens follow a threshold dose response curve. So that means you will be able to define a dose that's free of carcinogenic risk.
Yeah.
And typically, you can follow PDE approach, permitted daily exposure approach, and I think you should be able to define a limit.
Yeah. As your presentation clearly said that if there is a DNA reactivity, then the thresholds are not present. So this is probably mutagenic at any level.
Yes.
Okay, so in the case of the criteria for similarity, what is the minimum percentage to be matched with the reference structure? Is there any guidance on that topic that you could provide?
No, there is again same answer that I gave before. As far as I have seen so far, there is not a single guideline that tells us that this is the similarity value that has to be adhered to qualify as a surrogate or analog. And the reason simply is that first of all, there are all kinds of different similarity values or similarity measurements available. So every one of them is different from the other. And the other reason is that the whole structural similarity is not an indicative of the quality or the fitness of a particular surrogate. It depends on the kind of groups that are present around the alert. That's why we have defined a new type of similarity approach, which is called alert environment similarity, which ignores the rest of the compound at first. First, it checks into the the area around the alert, and if that is fine, then it checks the overall similarity, just to make it more concrete. But again, there is no particular value, absolute value that I could tell you that would make that would help you to find a particular analog. So basically, the similarity values for us at least, it's just a tool to give you the most similar analogs so that you can start your analysis. After that, the value should be forgotten and you should only look into around the alert itself. You are lucky if you get something similar, compounds or similar compound that has a similarity value above 0.7, say, and then it's fine. I mean, that's good. But you should not reject compound that is below a certain similarity value.
Okay, thanks, Suman. I'm going to bring someone off mute who would like to verbalize a question. Hello, Bijoy. Hello.
Yes. Hi. Thank you for the opportunity to ask the question. So my question is to Dr. Sebastian. In the case study that you showed, you actually showed that you have identified close to 8 analogs based on the data availability. So my question is, and I see how you selected the one that you needed to do the QSR was based on the TD50 values that were available and the most conservative one. But when I looked at the 4 structures, the analog 1 to 4, I could see that they are very similar, and maybe the analog 2 and analog 3 would have been a better choice, and they also had some Carci data as was mentioned in your slide. So why wasn't they considered together? I mean, maybe the analog 2 and 3 would have been, if we have done a benchmark dose model, they could have given us more conservative values. So was that checked, or just based on the TD50 values that you found in your analog data set, you went ahead and selected the analog?
Yeah, structure 2 and structure 3, in terms of a data quality, it was of not up to the mark, both in terms of the values and also in terms of the studies. And that's definitely one reason. And secondly, the chain length, both on R1 and R2, we wanted to be more closer to our impurity, C2 and C3. So these are the 2 reasons actually why we went ahead with structure 4.
Okay. All right. And just a second question to that on the topic on nitrosamines evaluation, right? So is there any consensus on the use of the toxic equivalency factor approach, or the TEF, or the potency equivalency approach for these class of compounds?
You mean you are talking about potency, cancer potency?
No, the toxic equivalency factor approach, commonly used for class of compounds like that are known to be carcinogenic.
Yeah, to my understanding, even the regulators also are actually following the approach that's stipulated in the ICH M7, and something similar to what I demonstrated in the case study. And I do not have much further information actually on the toxic equivalency, which you mentioned.
Okay. Yeah. Thank you.
Okay, I have another question from Osan. Hello, can you hear us? Hi, Osan.
Okay, thank you. Yeah, my first thank you. My first question is, would you share the slides after the presentation?
Yes, I can share the slides. Absolutely.
Thank you. My second question, if you don't mind, is Sebastian mentioned that for clastogenicity, it is possible to calculate, which is correct. But when you have like very limited data, for example, you have just 2 weeks data, how do you calculate in that situation? Do you use the 2 weeks data to calculate the acceptable limit?
There are a lot of disturbance, so I only hear in between. So could you please
Okay, I can repeat the question. So I think my question is in follow-up to your response to one of the questions, and you mentioned that if a compound is, for example, non-mutagenic or classogenic, and then because classogenicity, as you know, is assumed that there's a threshold. So, and you rightly said that then it's possible to calculate. My question is, when you have limited data, I mean, the data you have is, for example, just 2 weeks data, can you still use that to calculate? And if you can't, what do you suggest in that kind of situation?
You were talking about 2 weeks data, 2 week
Yeah,
study
for
of
example. Yeah, for example, we typically calculate, I know, I'm not saying that is the norm, but we typically calculate when it's from 28 days study, but sometimes you just have, classigenicity data from in vivo study of 7 days or 2 weeks. And then my question is, do you calculate using this data? And if you don't, if that's not your recommendation, what would you suggest in this type of situation?
Okay. Yeah, for clastogenicity as an endpoint to serve as a point of departure to derive a permitted daily exposure, that's can be considered. It's an acceptable approach, provided that you are sure that the non-threshold related mechanism is not operating for that compound, and the carcinogenesis is likely to be because of clastogenicity and not intercalation, a DNA, or or such mechanism. So in such situation, you can use the clastogenicity data from a typical ICH guideline studies. That not in most of the situation it's maybe a two-day dosing study, or it may be a part of a repeated dose toxicology study of a 14-day duration or a 28-day duration. So you can very well use those data and derive value. There's no need that it has to be of a chronic duration.
Okay, even to derive for example, a permitted daily exposure limit, which is meant for potentially chronic exposure. So you mean that even a 3-day study, a classigenicity study, can be used to calculate an acceptable daily limit for a lifetime exposure?
That's right. So in that situation, you need to ensure that the value that you derived based on the clastogenicity data is going to be protective of non-clastogenic effects also. So
but how do you know that? How do you know that when you only have the clastogenicity study, you don't have data from any other, for example, general toxicity study? You only have genotoxicity study.
Yeah, so that's where there are, there in such situations there are different approaches that one can adopt, and there is no single approach will solve the issue. Lack of such data is really a challenge. You may go for again a surrogate approach to not really looking about clastogenicity, but looking about what's its potential to cause target organ toxicity, or what's its potential to cause teratogenicity, etc. Again, you may adopt a surrogate approach. That's certainly one approach. And in some case situations, you may also think of a default TTC approaches for non-cancer endpoints. It may be a Kramer classification or other default TTC, depending upon the context to which you are deriving these values. And certainly it's not going to be straightforward. There are a lot of challenges involved.
Okay, thank you.
Okay, Jadip has his hand raised. I'm going to go ahead and take him off mute for another question. Hello?
Hello.
Jadip. Hi.
Hi, thank you for this pleasure and all. The question is basically to Sebastian, sir. So you have shared the slide. It's a really wonderful slide. So I have basically two questions from the question is basically a mix of safety assessment as well as risk assessment. What have been going through recent literatures post the risk that was posed by nitrosamines. So basically, you have used TD50 value. I mean, the ICH M7 guideline says that we have to use TD50 value for deriving or the BMDL10 approach values, right? So we also have a concept called as margin of exposure value. This is basically a bit of difference in the exposure value. So in this margin of exposure, we have been recommended by the few research scientists in initial days, and later on the FDA also came up with this regulation that in MOE we use TD25 value if we don't have anything value with us, and in the next case, if we don't have a TD50 value, then we will go with BMDL 90 value also, wherein we have 90% confidence. In case we don't have anything, then the last approach is BMDL 10 value, what you have referred to. So my concern is that in this case you have used TD50. I mean, the ICH M7 uses TD50 value, and you have also taken TD50 value in deriving nitrosamines. So in TD25 value, what is the issue in taking TD25 value, and why can't we go with TD25 value? This is my first question.
Yeah, I suppose the thought process behind TD50 is it's actually a midpoint response, and in animal studies, for that matter, any studies, so there can be a lot of variations around the background, and probably that my thinking is that TD50 could be when you define that value, that's going to be more sure about its potency, rather than you define a value which is at the lower end or higher end of the dose response. Given the complexities in carcinogenicity study, because dose spacing and number of doses, there are always limitations, and probably that could be the reason why when you have a 50% response, maybe more reliable.
Sure, sir. Yes, sir. Another, sir, when you have answered this question, I'm very thankful to you. With this point only, I mean, the concern question next to this question related is that since you told me that TD50 value because of this reason what you have given, I read the FDA guidance document that has been prepared with the collaboration. Now EMEA is also participating, and there is a recent consensus that all these databases, they will be merged into one, and there will be only one single individual value, because different companies are coming up with different PD value, TD50 value as per their convenience. Sometimes they just do it to safeguard themselves from MHRA audit, because in Indian audit they have to do. So question is that if by default TD25 value is already 40 times overestimation of toxicity what we are doing for the risk assessment process. So if I concern being a safety assessor, not a risk assessor, my job is to collate all the data, and I'll put all the data into one substance, and I'll then mitigate the safety. Either I'll mitigate or I'll leverage the safety from the data available in literature, be it a risk assessor does over the website, or he keeps it, or I do from the risk assessment point of view, to conclude a final safety assessment from the cosmetic and personal care point of view, wherein I will be merging all the data in a single product case. So by default, your TD50 value, what ICH M7 has also referred in the CPTB database also, it is very high. I mean, even TD25 value is 40% times overestimation of the toxicity. So aren't we somewhere just trying to make the thing simple and lucid for us, just for the sake of doing the safety assessment or the risk assessment you call? And is there any way, or if somebody uses TD25 value to derive rather than TD50 value, which is anyways more conservative than the normal values, so is there any risk or benefit associated with it?
My comment is that we need to really understand the principle or background behind using TD20 or TD25 or
Right.
TD50.
Approach, what is the approach they have used basically?
If you are using TD25, that means it's a 1:4 risk level actually. TD50 is a 1:2 risk level. And when you extrapolate down to 1:1 lakh, this is not going to actually have much impact on the final value that you are deriving. Although if you are starting TD25, you may divide it by 25,000. And therefore, irrespective of what is the starting point, I don't think the final value is going to differ much.
Okay, sure. And this is the last question from my end is we have referred TTC value 1.5 microgram per kg per day is a default value. So there have been some of the compounds when we compare with the different in silico models, you call it an OECD QSAR toolbox or TTC, we find difference because I mean when I was going through all the 31 questions and the extended plugins also, there was question number 19 and 20 that actually creates difference in the alkylation judgment of the OECD QSAR toolbox and TTC. So when I have even encountered some of the polymers and the compounds, they were declared safe because they are not getting absorbed in the body. Technically, they are not getting absorbed, but because of data shortage, I had ran them all over those all over those to do softwares. Now the question is, after running on the two software in the TTC, I found it to be clearing, and the OECD QSAR toolbox, I found it to have a toxic signal. So in this case, for example, if I want to derive a PDE for that, or if I want to use the value of TTC, what we have told, in case of safety assessment, I have to divide the margin of say in margin of safety, I divide the NOEL with the all the factors and all SET, the systemic exposure to. So in this case, sir, what should be my approach? Because both softwares are giving, one is giving safe, other is giving me a genotoxic positive alert.
Yeah, more of an expert inputs probably needed. You know, relying instead of relying only on the tool output, probably our in cerebro, inputs
Right.
probably. That's my thinking.
Okay, sure, sir. Thank you, sir. Thank you for precious time and allowing me to resolve my doubts. Thank you for the admin for this help also.
Okay. So we have quite a few questions. So this is not exactly related, but someone's asking how QSAR can help analyze steroid hormones.
Okay, so this is a good question. I have in our software we have quite a few steroids, and we use them regularly, and we understand that they have a slightly different mechanism of action, but they are handled as any other molecule in the QSAR software. The reason is that our software, or not only our, but any QSAR software that is used in this area of toxicity measurement or toxicity assessment rather, they can handle multiple different types of mechanisms. So it includes compounds that can cause mutagenicity by, say, alkylation or say intercalation, or like doing something which is not really mutagenic but genotoxic, like steroids. So steroids are handled as any other molecule.
Okay, so someone's asking if an in silico outcome is positive, but a closely similar structure is negative in the literature. How do you balance those two conclusions?
Yeah, so that's the whole activity that we do during the, or not we do, but we recommend doing during the expert review, and that actually happens quite often. That the software gives you one outcome, and that could be positive, but then you find out that you have a analog which is very close, and that is negative. So that should be taken into consideration. Usually that type of consideration is already given by the software itself. Our software includes this kind of tools that it takes into consideration the closely related analogs, and modify the outcome that is obtained through the QSAR directly. So that happens all the time. And one of the best examples is, suppose you are assessing an impurity, and you find that the QSAR software gives a positive outcome, but then the API contains the same alert in the same type of alert environment. So API can also be considered as a very closely similar analogs. And as you know, that API are already a known non-mutagen. So this is the same example. So this falls under the expert review process.
Sure. Thank you.
Sure.
Another question is, have you come across any nitroso compounds and reported as not cohort of concern? If yes, what is the recommendation for control strategy?
Sebastian, maybe you are better suited for this kind of.
Yeah, I haven't come across, personally, but I understand that there are compounds. There are examples of similar, scenario, but yeah, not really, encountered it actually. So I don't have anything more to comment at this, at this point. But I think, my thinking is that, in those situations when you have a compound specific data, I think, that answers everything rather than, the class effect. When you say nitrosamine, your risk is considered high based on the class effect. However, if a reliable compound specific data that says otherwise, I think there is no reason why you should not believe the compound specific data.
Yes.
Okay, so I think we are coming to the end. Most every question has been answered. And let me just double check and make sure that no one's hand is raised. I don't think so. So I think we can go ahead and end this session. Of course, if anyone has any follow-up questions, just feel free to reach out to us directly. You can reach out to me and I will go ahead and forward your question to either Sebastian or Alejandra. And I'd like to thank Sebastian for taking the time to be here to answer questions today. So thank you very much for the wonderful presentation and for your
Thank you,
time.
Sebastian.
Thank you.
Thanks a lot.
Thank you, everyone.
Yeah, so like I said at the beginning of this session, our final part 3 of this webinar series will take place on August 4th. And in that session, we plan to discuss advanced concepts and applications for M7 assessment. And there will also be 2 presentations during that session. One, Aldo Kalpi of Amgen will be presenting with industry case studies, along with our Dr. Rustam Sayakov, who's present in today's session from MultiCASE, who will be discussing it from our end. So hopefully you'll be able to attend, and again, reach out with any questions in the meantime via email. So thanks again, everyone. Have a great evening, day, wherever you are in the world, and we'll come back together again on August 4th.
Thank you, everybody.
Thank you. Bye.
Bye-bye.